Got it. Okay. Okay, so I welcome you all for this interesting talk, translation research from basic foundation, a personal journey by Professor Upendra Nongthomba. A little bit of introduction about the speaker. He's a professor and chair in the Department of Biomedical Genetics Laboratory, Developmental and Biomedical Genetics Laboratory, and the department is Department of Developmental Biology and Genetics. Okay, so he has done his PhD from University of Mysore on the development, on the genetic analysis of indirect flight muscle development in Drosophila, and followed by his postdoc from University of York, which is one of the very accomplished and big universities, from 1998 to 2004, and then from 2004 onwards, he's at the institute as faculty, as assistant professor, then associate, then present professor, and now he's the chair. So, and the type of research that he does is really, really amazing. He tries to, understand the developmental processes as well as neurodegeneration, myopathies, in model organisms such as Drosophila, zebrafish. These are the model organisms. So, today is going to talk also about translational research. Okay. So I'll just invite him and ask him to really go ahead with this wonderful talk. We're looking forward to. So, please.
Okay. First of all, thanks, Partha, for inviting me and asking me to just talk to you guys. But I don't know how many of you are biologists. I mean, at least some done biology? No, right? None of you. 1, 2 of you. Biotech. Okay. So I will he asked me to talk developmental biology. I said that no way that engineering people will be interested in developmental biology. Instead, I wanted to share, just by doing basic research, which we do most of the time, right? At least in the biology, most of us, we do basic research actually, right? Of course, during the grant writing, we will just put that we are going to cure cancer or something else. But take it from me, so far, at least from India, we have not been able to cure any of the disease, right? But people have actually, including PI from this institute. I'm not talking about negative way, but people have taken lots of money, public money. They have become professor, they have got awards, rewards, they have become director, but nothing has happened so, right? Okay, so that is where I thought that, we do basic research. My group is interested in muscle diseases or neurological disorders, but where we can make a difference, that was the case, okay? And knowing basic research, basic research means, let me just define you what is basic research. I don't know why it is not working. Basic research, we know that any way you are trying to understand the natural phenomenon, right? For example, say how you have become what you are today. For example, if you are trying to understand why you have only 2 ears, 1 nose and 1 mouth, or something very basic thing, right? Trying to understand it, to unravel the whole thing. This is the basic research, because you require to understand the mechanistic detail at what time of development, for example, you got the hair cells to be different from, say, other cells. These are basic research. Any natural phenomenon. So if you are trying to understand what is there in at the electron level of the neutron or proton, and today we know that every element, right? Finally, after the quark, anti-quarks, and everything, is just some string playing some kind of music, and that is why we are here, right? So that kind of thing, trying to understand the phenomenon, is the basic research. But if you are trying to intervene this basic research, that is what we call as a translational research, right? If we know that there's something abnormal goes and caused a disease, if we can intervene it by different way, that is a translational research. For example, say if you have a cancer, if you know the pathway, for example, now the present cancer drug is on inhibiting a signaling pathway called as the GPCR. Okay, yeah. So you can design it, right? So people used to think that you should do applied research, means that everything should be on the apply or translational impact, whereas basic is quite different. But today we know that these are just two sides of the same coin. Okay, so one classical example is that how you think depends on it, and you know it. This I am just putting it just to impress upon that I'm not going to talk only about the biology, right? Right? So Albert Einstein's equation, I mean, if you look at it, he was just interested in the how energy form and from the say simple atom and things, right? But the people, those who knew the things, I mean, they changed it to atom bomb, right? And today, actually, we have a hydrogen bomb and atom bomb, which can actually destroy the Earth almost like 100 times. But nobody is using it because people just want to have a peace that I have atom bomb, you can't attack me, right? I also put it, right? We all will destroy, right? This is kind of a peace deal which people have been doing. So there are useful things, and some of the things people use it. For example, fusion technology. Today we know that fusion technology can produce energy as bright as the sun itself, right? Okay, so these are something which, if you know it, then only it can happen, right? A simple hairpin, for example, some of you are using, can cause almost like 10 times that the energy which the Nagasaki, Hiroshima bomb, atom bomb, right? But we have the technology to do it? Actually, we don't have it, right? Okay, but they have energy to produce that much, right? So simply coming from directly from the physics to biology, if you look at the drug discovery, Alexander Fleming, because of him, many of the things now we are able to survive. Remember that till 1960s, 1930 to 60, India's average age, right? Okay, was just only around say hovering between say 38 to 40. Today, our longevity age is almost 68. I mean, it's thanks to all this discovery, right? But that time he discovered just a drug which can kill, right, bacteria. And today, you are killing the susceptible bacteria. So susceptible bacteria will be killed. Then the bacteria also have the thing that they will try to defend themselves and grow. And these are what we call as the antibiotic resistant bacteria and things. And because of the basic research, now people are coming that the antibiotic alone cannot kill the bacteria. So we have to develop other things which can kill the bacteria. Okay? And all these things today, you can do it just by having the AI. People are using AI to look at the all possible target in the bacteria. Every protein, genes, small peptide, everything can be looked at, and you can design drugs, right? There are at least in India also we have one, but there is one company in America, they are $1 billion, right? Okay, just designing drugs based on the AI training, right? One of the best things which happen over time today is that you know about this drug, right, which can save a person, but it costs 16 crores, right? You might have heard about this drug, right? Okay, yeah. If you get bored, just hands up, then I will narrate you some more stories, not politics though, because Partha warned me that you are not going to talk nothing but science, right, Partha? Okay, yeah. Okay, so this happened because of the basic things. So there is a gene, what we call as SMA, right? Okay, spinal muscular atrophy disease. Usually most of the protein are named based on the disease that they cause. All right. So there is this gene, if there is a splicing, let's not worry about it. So if you don't have this, what we call as the completely proteins are not formed at all, then the individual will not survive more than say 2 to 3 years. Okay. Then there are a mutation where they can survive. You know, the protein, it can be abnormal, or the protein is not efficiently working, right? Okay, so this drug was designed, Nongthomba was made so that the protein, where protein is not produced, protein will start producing, right? It is based on the adenovirus. Adenovirus is a kind of virus which doesn't cause any adverse reaction, right? So and this gene is very small, so you can put it and you express it one-time injection, the individual will get cure. So far in India, 6 people got it. Out of 6, 5 got almost cure, and the one died outright. Okay, so we can't guarantee that everybody will survive, right? We don't know what made that child die. Okay, so this happened because from 1960 onward, people knew when does the SMA protein require for the development. Okay? That basic thing was not known. The gene was not cloned and things. Then it would have been imported, right? So you have a SMA 1, 2, 3, 4 category based on the, as I told you, based on the phenotype, in how bad the phenotype will be. Phenotype means that what was finally visible to be. Okay. So there are other SMA patients. They can take this. This is a small molecule inhibitor, and they can use it. Again, remember, unlike 16 crores, this actually goes around. You can see it around 12 lakhs per bottle, right? Okay, and you need close to around 3 bottles per year, and it allows him to survive. The motor neurons I mean, this, if you take it, it will allow the motor neuron to survive longer. Okay, yeah, and so at the moment there is a buy 2 get 1 free going on, right? Okay, so because they know that in India, if you say free, then only Indians will buy, no? So they have this offer for Indians, right? Okay, so what we have been doing is that we have been working extensively on remember, we know today how, say, a black hole, which is almost like, say, 57 million years, light year, I mean, away from Earth, how they look like and how that is. I think it is called M57 or something. Yeah, we know about it, but actually we don't know how our cells are formed. Really, take it from it, right? And biologists thought that we know everything. Then now 5 PI in IAC, they work on actually physical property of the cell membrane itself, including a physics person from Prerna from, physics department. She works on the understanding the mechanical aspect of the cell membrane itself. And the more we thought that we know about the cells, actually we don't know. People used to think that, okay, there is a RNA, DNA, all these ribosomes and all things. Today, there is more. In the RNA itself, there are thousands of, at least few tens of RNAs, different variety, and we don't know what they do. Then we thought that, we know RNA, then there are something called as circular RNA, circular DNA. The more you get into it, there is a more problem, which we don't know, right? Okay, so this is where we get in, because any problem during the development of the muscle will lead to something called as the muscle diseases or myopathies. And neuron, same way, neurons. The good thing about the muscle and the neuron is that, muscle cannot survive without the neurons. Neurons cannot survive actually without the target muscles. Okay. So the crosstalk between two are very important. And for the aging, because we work on the board, and we got into the aging because today what we know is that the aging is nothing but the muscles and the neuron as we age, just moving little bit. So the neurons on the muscles moving like this, little bit of moving as we age, we become weak, right? Okay. And not only that, if you look at the diabetes or the cancer, so or say tuberculosis, any kind of disease, people die because of the weak muscles and the neurons. Okay, for example, say cancer people die because of the adverse effect of cancer called as a cachexia, right? Cachexia is a muscle disorder, muscle mass, loss of muscle mass. Okay, right? And 25% of the cancer patients die from the cachexia, right? And which kills the most in cancer? You know anything? Actually, 35% of the cancer patients die from what you do for the treatment of the cancer? Chemotherapy, right? So 35% of the patients actually die from the chemotherapy and the radiotherapy. Okay, right? So they actually are not killed by cancer itself, the cell proliferation that we talk about, right? Okay, so this is what we look at aging, how we can intervene it so that we can have a healthy aging, right? Okay, so there are many things, CRISPR-Cas, for example, you know it, basic research. If we didn't know about the basic research of the CRISPR-Cas, right? Remember that this is where I'm talking about that. 1979, this was discovered, right? CRISPR and the Cas9, all these things were discovered in the 1980s. And in India, we didn't do anything. It is not that Indian people who are not working with the bacteria, but we never saw the opportunity, my point is. Only when the people from outside, they started, that is where we start following. The same thing applies to your gut microbiota. Today, every paper you take it, we also published it only a few days back about how gut microbiota controls the neurological disorder, okay? I'm blaming myself also because it was Charaka Samhya who told almost like 4,000 years back that you need to have your gut and the liver properly. If you keep it, you won't suffer from any disorder. Okay, but we never studied till 2012 when the Poland lady said that actually the bacteria present in your gut actually decides your health condition. From 2012, but 4,000 we didn't do anything. Okay, and today there are so many labs in India itself. They are working on this. Right? Okay, so if you look at it from 2012, there are thousands of papers on. So I don't know what happened with Indian. That's my point. We always follow. We don't even look at what existing data or the existing knowledge, right? Okay. So in this direction, these are the CRISPR-Cas thing people are looking at. Today, hemophilia, sickle cell, these all are now available, FDA-approved CRISPR-Cas therapy. Okay. They are not very expensive. If you have any friends and things, these are not available yet in India, but they can go to America, get cured. All right. Okay. So my personal thing is that whatever we are doing, how we can, with that background, I just want to share what we have been doing. Okay. Right. So we were talking about this vaccine, right? Okay. Because of remember, today we are talking about because of the COVID-19, right? Yeah, vaccines. In developed country, however, the vaccines are stored in a very good way. But in the developing countries, you need a cheap way. Developing countries or the African underdeveloping countries, all these places, you need cheap chemical which you can store it, right? When we look at it, actually, we found that this chemical, you can read it, thimerosal is used extensively. Okay, it is an organic mercury, right? Organic mercury used for, because it doesn't allow any microbe to grow. Simple as that. But when we look at it, by the way, this is available for 160 rupees per piece. That's it, right? So that is why this is extensively used in the many food storage and things, right? Okay? Without actually knowing the adverse effect of it. Okay? And when we look at it, we didn't find the exact mechanism for the thimerosal. It is toxic to the microbe, we know it, but what are the toxicity level? That is where we did all the toxicity things in using zebrafish as a model, because zebrafish is a very good vertebral model, right? Good thing about using model organism is that, without getting into too detail, is that you can actually dissect the mechanism. And we found that actually thimerosal actually act at the mitochondria. Okay, yeah, mitochondria in the eukaryotes. Okay, right? Now, it works on the mitochondria. We knew the mechanism, but bacteria, they don't have the mitochondria. Then how they get there? So it is because of the genotoxicity. We call it as the mercury getting into it and acting on the genome rather than mitochondria. So in other words, if we make this organic mercury not to enter the genome, then it should be fine. And that is where we had a very simple thing. Most of this organic compound, they can take with something called as the cysteine bond, cysteine, right? So we use cysteine, right? Cysteine again is very cheap, right? 1:1 ratio, and we have studied. Being in IIC, of course, you wanted to get something which we can do, is that we can perform all the mechanism, right? With the CD and all this technique, we use it to look at it, how they are forming the conjugate and everything we have done it. The best part is the once we have this conjugate, the organic thimerosal with the cysteine, it doesn't kill the zebrafish. Okay. So the killing toxicity level just go down like anything, right? So it's not that toxic to the eukaryote because it is not targeting the mitochondria because of the presence of the cysteine. Okay. And the other thing is that, however, it retains the bacterial killing affinity, what we call as the CFE. It can kill efficiently, right? So the tomorrow, we haven't patented it, but tomorrow it means that you just mix the cysteine with the thimerosal, and they can be used for any kind of storage, vaccine storage and things, right? Okay, don't sleep yet. Yeah, right. So this is a very cheap way. When together, if you look at it, for example, cysteine also is very cheap, around 250, 280 rupees. So then you just need to mix them, and they can be used as a storage thing, so that there should not be any microbial growth. Simple as that. So tomorrow any industry can just take it out, right? Translation, right? So then what we have done is that we have been working on the, as I told you, the bacterial infection. What insects produce is what we call as the antimicrobial peptide, which is the kind of innate immunity against things, right? You see, insect, any organism, they have to survive, right? So they get infected, they will produce something called as antimicrobial peptides. And we have been studying the mechanism of how antimicrobials are produced in Drosophila for a long time, right? But what we realize is that in Drosophila, there is a very potent antimicrobial peptide called as the drosomycin, okay, which can kill both fungals, bacteria. So when we look at it, silkworm, they don't have it. And you know that India is the second largest producer of the silk, and many of these silk, right, in India, they suffer from all these fungals and the bacterial infections. So what we thought was that why don't we generate transgenic silkworm with this antimicrobial peptide, drosomycin from Drosophila. Because the silkworm, somehow they don't have this antimicrobial peptide, right? So what we did is we produced this transgenically, the silkworm, and you can see that, let's not get into it. You can see all this green is because of the having those antimicrobial peptides. Okay, we take it so that we can follow that they have the antimicrobial peptide, right? So good thing is that they should be disease resistant. Yes, these are all experiments to prove that the vector is there. You see 60%. Where the infection, first one, just goes almost 60, 40 to 60% resistant, right? Not only that, it doesn't cause the insertion of the drosomycin doesn't compromise any of the productivity. So the tomorrow it is now transferred to the Central Silk Laboratory, SBRL, here in Bangalore, and the Central Silk Board, they can actually use it. I mean, right? Because silkworm is such a insect that they don't fly much. So there is no danger of contaminating with the wild population. Okay, right? They survive only for 3 days. They don't fly very far, right? Okay, so they can use it directly, and they don't compromise. If you look at our transgenic cells, they don't compromise in any productivity or anything. Is that okay? Yeah. So the simple thing what we were doing in Drosophila, you see, remember in India, there are so many labs working on silkworm, and I don't understand that why they don't think about it. Right? Yeah, I'm working in Drosophila. I was just an advisor. We call it as the RAC, Research Advisory Committee of the Central Silk Board. I went once, and I just saw it that they are not doing anything. They are wasting their money, right? So that is where I thought of this, right? Okay, and we have made it now. 3 years, 4 years, we have made actually three transgenic lines. One is overexpression of the other, like release and the other things, okay, which actually again increase the immunity. So it is all about just thinking. I mean, just change in the little bit of attitude, and you can make a difference, right? And similarly, we have been working with the doctors in the demands, and we generate, as I told you, all these defects, right? We look at it by using the Drosophila or zebrafish as a model, all right? So let's not get into too much. The good thing about the model system is that we can really dissect what kind of mutation it is, okay? In the sense that which kind of mutation you should be able to reverse it back by putting adenovirus or the protein expression system, if you have it, which one you will be able to recover, right? Okay, those we can just predict it by making mutation in Drosophila within, say, 3 months time, because Drosophila transgenic require only 3 months, right? Okay. Now, the good thing about having this thing is that, just like I told you, we have been collaborating with the people to just develop technique and the tool. This tool, we develop it. You can see that Raman, so far to diagnose muscle diseases, what you have to do is that you have to take the biopsy. Biopsy is at least 0.5 millimeter by, say, around 1.5 centimeter by. So 1.5 centimeter by 1.5 centimeter chunk of muscle you remove from the calf, and we do electron microscopy, immunohistochemistry, and everything to identify. But today I can tell you that you can just take a needle biopsy, and you can do Raman and IR, and you can actually tell that whether you are suffering from what kind of myopathy. And this, thanks to our Drosophila, as I told you, Drosophila muscle is a highly conserved tissue. So we have this myopathy condition, different kind of myopathy conditions, right? And we do the Raman and the IR, and we develop all this signature. The take-home message is that you look at it here. Whatever we have done in Drosophila, different thing, the same thing when we correlate with the Drosophila, your blue and the red. Drosophila and human, right? They go together. So the nemaline myopathy, Drosophila nemaline myopathy, go together with the human myopathy. And the good thing about the collaboration is that the Umapati's student, Rekha, never knew that actually we have given her human muscle as a sample to do the Raman, right? Okay, so it was a kind of blind experiment. Okay, and when she came up with all this data, so we could figure it out, right? Okay, so Raman and IR can be used easily with a needle biopsy, right, to figure out which kind of myopathy the individual is suffering from, right? Similarly, we developed, this is patented one, with Rudra Pratap. We developed something called as the Doppler vibrometer, laser Doppler vibrometer, LDV. LDV can detect based on this simple mechanism that you put a laser beam and look at the vibrations, right? So the vibration, wherever there is a vibration, it generates the vibration frequency, right? Increase, just like in your paper when you put it, right? Thin or the thick paper will generate different vibrations. That is what actually we have looked at it. And by looking at it, this vibration signature, we can figure out whether the individual is suffering from the cardiomyopathy, nemaline myopathy or not. Okay, so it's a simple thing. I mean, this is just, you need to think it. But remember, I didn't have this technology, right? Only what I had was that I have the model system. I approach the right people. So if you are desperate, then only you will approach the people. Desperate in the sense that if you have other thinking. If I was just like any of my colleagues, happy to publish with the, the model picture and thing, we would have not developed this technology. The good thing about IAC is that we have people expert in the different fields. How we can come together to work and, which should be useful to the patients or the people. That is what we need to think about, right? Okay. So similarly, we did with the simple electrical impulses, and we were again able to differentiate all the myopathy conditions by using simple electrical signature, right? Finally, when you have all these mutant conditions, then you can use it for the drug discovery, right? Okay. So the one of the most important how we started was that unlike neurodegenerative diseases and things where the patients come at the last, by the time they have already suffering from dementia and things, epilepsy patients, you see, dementia, once you have Alzheimer and things, now it is very difficult to bring them back to the normalcy. Okay? Parkinsonism, for example. When the patients come to the hospital, they have reached a stage where it is very difficult to intervene. But by the way, today, we know that Alzheimer can be predicted at least 17 years before the onset of the dementia. Okay? But epilepsy is something which is generally you can see among the population, right? And all the allopathic medicine available, drugs available, actually long-term intake of these drugs actually makes the individual dyskinesis, means that the movement problems and things, side effects. Okay, so if you look at the epilepsy data, 9 in the 1,000 individuals suffer from epilepsy in India. All right, particularly in Bangalore, surrounding to the Bangalore city, there are the frequency can go up to 16% per 1,000. Right now, we generated all this model in Drosophila. So the epilepsy, the epilepsy is that finally what you are going to see in the epilepsy is that is the normal. So the epileptic patients, they can have this kind of neurological readout. Okay, EEG will read out this. So we type of epilepsy depending on where the brain is affected or the genetic and everything. Let's not worry about it. Okay, so these are the major things, right? Okay, yeah. So what we wanted was that a drug actually or the natural compound which can inhibit the epilepsy condition. That's our measure, right? Because the existing drugs they don't address this completely, right? Okay. So if you are interested, I can talk about it later on, and you can, any one of you are interested, you can, if you have friend circle and things who are suffering from epilepsy, actually they can talk to me, right? Okay. But what we did was develop a model in Drosophila to address this thing, because finally the problem which we see is that the most of the existing drugs, they target only the last, that big arrows, okay, the phenotype. Just looking at the phenotype, not the early developmental thing. Okay, so what we did was that they make all the drugs, and we look at the natural thing where from we can screen them. And one of the things which we have been working extensively is pomegranate. Okay, and we have shown that how pomegranate works as a rasayan, right? And in the Ayurveda, already there is a text where it says that actually pomegranate can be given to neurologically defective individuals. Okay, it is useful, but we didn't know. Nobody knew that how the pomegranate works for the treatment of the neurological disorder. And when we look at the data, we found that actually pomegranate works in the many part of our body. Okay, so we use pomegranate. Look at the first in the Drosophila, and I'm not going to show all the data as I told you, right? But why we started with the Drosophila is that the Drosophila is a very good model. Almost, if you look at the basic mechanism of the neurological thing, Drosophila has the same kind of things that's like the human. Only thing is that they have less neurons or the connection, right? Okay, so we use Drosophila. If you look at the Drosophila adult, you can see just like the neurological disorder, actually, you should be able to see them abnormal. You see in the epileptic patients, right? Similarly, you can see Drosophila, epileptic Drosophila, you can see it, right? All this kind of, this is different, right? Okay, these are completely different mutations, right? Okay. So we provide pomegranate or pomegranate derived compounds, and we look at how they are recovered. And you can see at the larval stage, what you can see is that, for example, say in the normal larvae, they will move around, the chemically treated to generate the epileptic condition. You see that they can't move, right? And now one of the compound from the pomegranate is called the urolithin A. You can see that the thing. Now this chemical, this epileptic condition, now if you provide a urolithin A, now look at it here, it recovers, right? Okay. So the simple readout, right? And then of course you can quantify these details and everything, so that you can see that they recover, right? Now you can see all these things at the neuromuscular level, where the the signals and the muscle movement happens, right? And you can see that in the chemical treated one, this is abnormal compared to the normal ones. Then when you fit with the compound, they become normal, right? Okay, so you can study all the details, right? And this is what exactly we did it. We identified that something called as the urolithin A. Urolithin A is present in a very small amount in pomegranate per se. Once you take the pomegranate, the compound, the bacteria present in your gut, called as the firmicutes, they convert this compound in the pomegranate, ellagic acid and tannins, to urolithin A, B, C, D. Okay, out of them, urolithin A is the one which is useful to the epileptic patients, right? So that you can say that the epileptic patients should be able to take the pomegranate and they should be fine. But the problem is that many of the people suffering from the neurological disorders, they don't have this bacteria which can convert, or the 40% of the population itself, we don't have this bacteria in the high quantity, right? To convert this compound erythritanyl, for example, to erythric acid or erythritanyl to urolithin A. So the idea is that they should be given urolithin A. Now, if urolithin A, how they will reach to the brain, and this actually my one student Sefali Misra's 5 years. Okay. Then we look at the Drosophila, then we look at the mouse model, right? We generated in the mouse model that you inject the urolithin A within 30 minutes. It was all modified in our serum, in mouse serum, right? But when we look at the brain has the pure form of urine. Okay? So that it could pass the blood-brain barrier, right? So the urolithin can be given, and these are something called the preclinical studies. Okay, so we study preclinical study in mouse, and you can see that. Don't worry about all the things. Data we were able to recover the seizures, right, in the mouse model also. Something which work in Drosophila. Similarly, in the mouse model, we were able to do it, right? So the good thing now is that we do know that how it works. This is where we are a little bit different, and IAC people want it to be different, because feeding compound or the feeding pomegranate, anybody can do it, right? Yeah, even the university people can do it, right? Something which they can't do is this, right? How the compound works, how the compound enters the cells, right? Okay, that mechanism is important, and this is where I can't do. I'm not expert in that. So who are the expert now? That is where again IAC is really good place, because we spoke to Professor Tilaga in IPC, and he's a student, and they made this linker of our urolithin A, and we were able to pull down this linker. Okay, you can see it, right? Then we were able to pull down that by using the brain from the mice, that we were able to pull down where this compound binds to what kind of thing, and we identified something called as the voltage dependent anion channel, VDAC1, as the target. So that this is a channel which will take, right, which is responsible for a binding by that urolithin. And the key thing for us again, in the most of the epilepsy cells, people have shown that actually VDA1 is abnormal. So what it is doing is that inhibiting the VDA1. Okay, so this is the whole story, and excited about it because we were able to show again what we have shown from the mice. We went back to Drosophila and inhibited the VDA1 in the epileptic Drosophila, and it should show the similar phenotype, right? And we saw the same thing. Actually better with the urolithin A. Once you put the urolithin A, actually it was showing better, synergetic with the Nongthomba. The second last one. Right? Okay. So it is fantastic. And this is where today we have got this now. I am able to speak to you today because we have put it for the patent, right? Going for the Indian patent is applied, so we will be going for the prior patent thing is done already, so I can speak out and we can publish it. And this will be going for the US and the international patent in next 2-3 months. Okay, so this is where you need to just think differently. My point is there. And where we could not do, we should not hesitate to talk it to the people, those who are expert. The mice work, we could not do it. So whom we contacted? We contacted James Clement in the JNC, and my student went there. And JNC, it was very good that the James Clement allowed my student Sefali to work in his lab and completed the whole thing. Okay? So we should not hesitate to talk to other people who can help your work and science and take it to the next level. And that is what my message would be. Okay. So all in all, we were able, we are the only 8 lab in the world, actually, I mean, including Professor Tillager's work, who have been able to pull down the target protein by using the chemical linker. 8 lab in the world so far. Okay. So this is the exciting thing. That is what I'm talking about. Similarly, we have done identify plant, the Calabis hamiltoni, which can be used. Actually, this plant is used by people in Tumkur area as a pickle and things, right? And we identify the compound that this is the Western people want to know the active ingredient, right? All the time. So we were able to identify from the 4-hydroxyisophthalic acid as a compound which can actually inhibit the, which can act as a neuron suffer from either Alzheimer or Parkinson's. Okay, so we have, so having model organism helps it. And similarly, I told you the pomegranate, you should start taking pomegranate, because if you work in the lab for a long time, if you don't do exercise, pomegranate is a good way, it works. So the pomegranate actually improve everything, your immunity, your longevity, your health span, everything you can get it by just taking a single lobe of pomegranate. All right. And the pomegranate that we have worked is the bigger one, what we call as the Chikaballapur variety. Okay, there are many varieties, but the Chikaballapur variety, when you break it, you will have 5-6 lobes, right? 1 lobe is enough. And you can store it in your fridge. You can use it afterwards. Okay. And it works fantastic. Okay. What we have shown, if you look at that paper, is that it increases immunity, it increases. Only bad thing is that it also increases the rasayana means fertility, and the Indian being now the most populous country in the world, fertility, I'm not sure about it, right? But it increases, okay? But that is what we have shown it, right? Okay, and so you can see it, it increases, it reduces the stress, any kind of oxidative stress, anything happens in the body will be reduced. And we have work out the pathway, how it works by targeting a Fox head pathway. That is what we have done it, right? Having all these things as a model, I mean, my point is that you can work out the mechanism. But most importantly, remember, when 2014, when we published, before that, pomegranate per kg was 89 rupees. Within 3 months, because all our story went to the newspaper, in every possible newspaper in Karnataka, it went. In Kannada, in Times, everywhere it was there. You can read it actually, it is there. And you wouldn't believe it, within 3 months, the pomegranate per kg it raised to 129 rupees. And I went to one of the market to buy it, and I jokingly asked that, why it was just only 89 rupees to 3 months back, why it again? Oh, one guy who in only Kannada, right? Who said that in Kannada, scientists have proved that this is really useful, right? And they don't know who was the scientist, right? Yeah. So sometimes you can help. That's the whole idea, right? Okay. So we model all these disease conditions. The whole idea of the disease condition is that, for example, this we made it the disease ECTA1 because in India, as of today, doctors are asking this patient to take supplements, all right? Okay, amino acid supplement. And we were concerned about it. And we knew that the supplement should not work, right? Okay, and the kind of supplement was, and we made all these disease conditions, you can see it, and we tested the supplement, just like in the human condition, aggregates were more, protein aggregates were more, swimming escape and behavioral, everything was abnormal. But you see, most importantly, supplement all what people are living at the moment, doctors are living, right? So these are the simple things, right? Yeah, where we can actually help the doctors and other people normally that what should work and what will not work, right? And of course, we have just published a paper where we think that if for the dystrophic patients and these patients actually costamere glycosylation is abnormal and we should target them, right? How to target is different, which we haven't worked so far. But what I'm trying to say is that the basic foundation, basic research is very important if you want to particularly want to work in biological related translational research. Okay, and this is just few examples that I'm trying to give you, right? Okay, so the most important thing, however, what I'm trying to convey, last one is that if you remember that most of the work, neurological work, when we look at it, I don't know how many of you know about the universe 25x, right? Here, what John B. Calvin, what he has done is that he started experiment with only 4 mice, right? Just allow them to breathe in the case, right? Okay. After 25 generations, you will have so many mice, up to 2,200. During the whole process, as the number increases, for example, they go through lots of behavioral changes. For example, say the guys finally actually they become more of like a ladies. They like grooming themselves, right? Okay, and the women, I mean the mice, female mice, they become bigger in size and they don't like to mate, for example. Okay, they are domineering. Yeah. Okay. You correlate with the present scenarios, right? Okay. All these things actually, right? They do it. But what people have seen is that you increase that instead of the case, if you make it in the rat park, for example, experiment where they have more places to play around and things, actually many of this behavior didn't happen. Okay. So the social interactions, talking with other things is also very important, right? Okay. So the basic research can tell you what to do, what not to do, and things like that. So at the moment I brought this slide finally is because at the moment we are lacking this social interactions and so, right? Okay. We think that if you put on your mobile, right? This is also science for that. Okay? I'm just trying to, because Pratha knows that I don't get bothered something other than the science. So I just wanted to convey you that please cut down your screen time, right? Spend more time with your friends and the families, talk to them physically, interact with them, right? And that actually is the most important translational research from basic foundation. Because this experiment showed that the moment the society crumbles, right? Okay, nothing remains. They all die. Okay, things will change. Yeah. So just have a good time, cut down your screen time, and interact with your friends in a physical way, and spend more time. Okay. With this, I stop. Right. Yeah. Thank you very much. I'm on.
Yes, yes. What can I say? I mean, this is a great talk and a great emphasis on translational research. What I also noted that, and he's a good friend. So what a giant step he has made towards translational research. I mean, the amount of data, amount of research that you have shown. And I have not asked him, but tried to go and visit his lab. I mean, that would be a great inspiration for all of you.
By the way, nothing is there in mind.
Yeah, you can figure out how things work out, how he makes things up, how how the students work. How
no, I mean many labs have big things, right? But for me, I don't keep any of the things. I like to work with the minimum things because I believe in philosophy that whatever we are doing comes from public money, and we should not overspend something which can be done in the other labs, right? For example, if I have to do a high sophisticated imaging today, I will approach him and get it done here rather than have another licensed microscopy in my lab. Okay? So that's what we should.
Yeah.
But please, you are most welcome to come and talk to me, but don't come to see I can do equipments or things like that.
So the talk, I would say, is a treasure as far as disease biology is concerned, and this will be one of the topmost talk in lab visits. There are other talks, but I see this as one of the topmost talks.
Yeah, he's saying because
and yeah, See, and it looks like collaboration is the key now.
Yeah.
That's what I gained from this talk. Upendra, I have a couple of questions, and then we can go to others also who might have other questions. So, what do you think? How this AI-based drug design? This is started up, but how accurate it is? Like,
The accuracy is something which we have to do with experiments. You see, the AI will only look at for example, say we also use the AI because my one of my students has got a company called the CCAT. If you know it, you can find out. It is called the CKAT. So they also use the AI. So you can predict is that at the level of this thing, this amino acid, for example, say in the urolithin A and the VDEC interactions, we were able to predict what are the important regions which are important for the VDEC and the urolithin to come and bind. Tomorrow, we are trying to make, say, for example, with the Tilalo, we already have a grant to make the water-soluble, highly bioavailable version of urolithin A. Okay. So how that will bind when modified, right? All can be predicted. This all can be predicted, but in real, when you do the experiments, because the VDEC is part of the membrane, for example, right? Okay. So the real experiment will tell that when it is in membrane, which one is the most efficient. So the prediction can narrow down how many, instead of doing 100 experiments, maybe 10 or 5, which one you have to do it, but still you will require the actual experiment to do it, validate it.
Okay, the second thing that I want to ask, Upendra, is that this urolithin A, how it works on larger mammals? Like you talked about rats. So did you go beyond this? Like what do you expect?
No, we have a preclinical study. Mice is a separate preclinical. That is why we haven't done it. The good thing about the urolithin A is already urolithin A is approved by the FDA as a food supplement. Okay, for another company who has applied for the patent, or they have it, not for the epilepsy, right? And their receptor is completely different, which work through the gut metabolites. Okay, so you can take it. Actually, you should be able to see urolithin in your. If you have the normal, as I told you, the bacteria, firmicutes are there, then you should be able to see urolithin in your blood within 30 minutes of taking pomegranates, right? Okay, but you can take the urolithin directly also, and you should be able to see it, right? Okay, it works. How much you have to take it? Up to 450 grams. They have done it. This company, Amatezan, they have done it, and they don't have any toxicity. That's the good part. So urolithin is not toxic to the individual. So it can be taken as a food supplement, and FDA already has approved it for the human consumption as a food supplement.
Okay, just a supplementary question. If I have to look at cellular level, which organelle that is a target?
Target is that almost every cell. And already last month, there is a paper which says that it improved the muscles, for example.
So is it mitochondria?
No, it works through the channels, so that it will have the different tissues differently, right? Okay. But at least in the neuron, what we have shown is that the VDEC channel is abnormal in the most of the epileptic patients, or most of the neurological disorder, like say Alzheimer and Parkinson. We know that these channels are abnormal. So at least urolithin will work in the neurons through the VDEC, right? That is our finding, and we believe in that. But there are, for example, say aryl receptor, another receptor for the gut. So that it has to be tissue specific, but urolithin will work for the every tissue.
Okay, so I will just move forward. If anyone of you want to ask, we have the person here. So if you want to ask something, go ahead. Anyone?
Yes. So there are different ways. For example, there are cold stress where you give in a specific cold temperature, then they will stop moving, right? But some will be in that cold stress also, some will be resistant to that, so they will keep on moving, right? So it depends on what kind of stress you want to induce into. For example, we do the heat stress, for example, you heat it in 37, most of them become abnormal or die. Temperature stress 65 degrees, you give it within few minutes, most of the fly will die, but the one which are resistant, they will survive for longer time. Your walking, flight, survival, everything. Something which is measurable. Yeah, anything.
Yeah.
Exactly. Temperature resistant to temperature. So you can measure behavioral wise, we have it. Behavioral courses to some say behavioral flight to walking. These all, if you have a stress in the neurons, directly affect the behavior. You can study that. The physiologically, you can study the survival of the individuals, right, in the specific stress condition, whether you can induce them in the chemical or the temperature. For example, we use the hydrogen peroxide, for example. So they will generate reactive oxygen species. So some of these, the one which are fed with the pomegranate, for example, they are resistant. So we are just looking at the measurable.
Yeah. Anyone else?
No, survival is one measure if you want to. You can look at the, for example, behavior of the food intake. Yes, so the fly is more stressed. For example, they stop taking fruit, right? Okay. We have many things. This is where the translational is. Absolutely, you can measure. I can tell you that today, for example, we have data where you put a one chemical in the less concentration, it shows hyperactivity. Whereas in the higher concentration, it shows hypoactivity. And we don't know what is the mechanism. This is the kind of thing that is what we are looking at. Because if you look at our paper, you will find that many of the things, like, for example, people have used for the treatment of the cancer, right? Okay, we have shown that some of these could be toxic to the liver and things like that, right? Okay, side effect also, right? Okay. So all these things can be studied using the model organism, and you can say that which one works. For simple thing, rasayana, there are close to 150 different way of making amlaki rasayana. Okay, gooseberry rasayana, which is Chyawanprash, one of them, right? Okay. So but there are 150 varieties. Chyawanprash or the amlaki rasayana can be made only by 4 things: gooseberry, right? Honey, ghee, right? All these things, if you put it in proposal, right? And honey, ghee, and Chyawanprash, gooseberry, if you put them properly, you can make a Chyawanprash. Yeah, but now you have multiple additions. But which one actually will be used? We tested 19 of them. I can't reveal you here because this is a public forum. But only 4 worked. 4 were not killing the Drosophila. All other kill the Drosophila. It means that something is killing Drosophila. Actually, they can be harmful for the human being also, right? So these are the very basic thing. Actually, you can do it using model only. It doesn't require much time, but at the same time, you can figure out what is going wrong. Okay. Yeah.
Sorry. Do we have any online questions?
No, sir.
Yeah. So rasayana, pomegranate, amla, all these are rasayana. Rasayana means, in complete sense, revitalize the whole body. So immunity, longevity, stress, everything, it works. And in that paper, what we have shown is that the pomegranate is actually, as it was mentioned in the Ayurveda text, is a rasayana. And all the rasayana parameters, whatever we could do it, we did it in Drosophila and it worked. Yeah. FOXO pathway, FOCAT pathway, mechanistic. So that finally culminates to a transcription factor, which is what we call as the FOCAT pathway. And that actually getting activated allows all the physiological things, at least molecular level. But remember, whatever said and done, even though you said it at the cellular level, just like most of the Western people want to know that mechanistically, but organism-wise, I mean, you know, Ayurveda works at the organism level, not at the cellular level. Okay? Yeah. Good. So, yeah. 35% of the, yeah, cancer death. Fantastic. You come and discover it. That is what we all are dying for in the cancer. So immunotherapy, targeted immunotherapy for the most of the cancer now is available. So it is up to you. I mean, that is why if the moment, if you know it, I can tell you that at least like 15 labs in the ISA will go bankrupt, right? They will, they will not have anything to do with it again. Right? Yeah. Yeah, silkworm. Right. Drosophila gene from the antimicrobial. No, I show you. It doesn't compromise any of the quality of the silk. It just prevents the infections of the bacteria and microsporidium particularly, and fungal infection is reduced, that's all. It is resistant. So the bacteria will infect these silkworms. Because of that, mass silkworm will die, right, in the larval stage. Today, because of our drosomycin overproduction, those silkworm will not die. Yeah. Yeah. No, plant, not plant. It is made in the worm. So the worm, we are making it transgenic in the worm, transgenic silkworm. So the most of the, you are correct, most of the plant and all these things will have this fungal and things because of their conjunction, right? Okay. If they don't have a good immunity, they will die. So the mass larval or the silkworm death happens in India because of this. But now what I'm trying to say is that because of this transgenic, which doesn't finally compromise on the silk productivity, at least what we have measured, but it is resistant to most of this fungal, bacterial, and protozoal infection. Because this drosomycin is a what we call wide-scale antimicrobial factor. So it really works. So it works against the bacteria. So that tomorrow, I mean, 60–70% of your worms are surviving without the infection. So your productivity will increase. Yes, survival of the worms. That's what I mean. So I'm the one of the first who became I'm the only one. I'm the only one who is I'm adjunct faculty to a alternative university. We call it a transdisciplinary university, which is around 8 kilometers from IS. I'm adjunct faculty to that place. it started with collaboration, as I said. I always believe in natural or natural derived formulation. Okay. I read, I think I may have it. If I want to use, I will show you that I came across this famous doctor's quote. And this changed my life. So as I said, your motivation can come from anything, anywhere, and you should be open to. And the moment James Benny said that, whatever happening, allopathy, right? Can't recapitulate whatever you can get it from the plant product. This changed my life. So in 2009, I was looking for the institute and thing where properly local health traditions are being studied. And this institute, if you want to please visit it, it's a fantastic place. We have all the local health traditions scientifically documented. We have the database of this, more than 6,800 formulations. We have it, and they also have, people translating it from the very old texts, those which are written on the pamphlet or whatever things, right? Okay, you should visit this institute. It is only around 8-9 kilometers from this place in Yelahanka. It is called now as a TDU, Transdisciplinary University. But it's a fantastic place. But you should not just look at the website. You should actually visit the institute, look at the database, their herbarium, and all these things you should see. And that is where I started collaborating with Professor Padma Venkatesh. Padma is now dean in the this petroleum university in Yarrahu. I think some years. But I started in that institute with her. At the moment, I am the only adjunct faculty from IAC working here. I have taken our even director, not present director, previous directors, and we have had MOU with them. We had PhDs, joint PhD students. So we have many things going on, but this is where I started my natural things. And now today, we have data on Ayurveda formulation or the local health formulation, actually, which someday I will show you. I can't discuss at the moment because that actually can cure, for example, ALS. There is a disease, neurological disorder. There are muscle disorder. The fly, we have tested in fly by making these human diseases. The flies which were dying in 5 days now survive till around, say, 25, 30 days. Right? So these are amazing. But mechanism we were not knowing it. Today we can tell because of the fly work that how these formulations work, what improvement they can make it, and all these mechanism, right? That can be understood by using this model of it. That is what I do.
I think this discussion will go on.
Yeah.
So it's already 1.5 hours.
Yeah.
Okay. So I'm happy that this has happened, and I thank Upendra. Let us.
Thank you.
Thank you. Thanks.
Okay. But not for them. But you think about it. What are the, you have